The natural world is losing its color. Climate shifts and habitat loss are erasing the ecosystems that support global plant life. Inside the Borealis station, the environment is different. Thick titanium walls seal off the cryobotanic vault from the outside world, creating an atmosphere that feels heavy with the weight of centuries. This facility was designed to act as a secure loophole against extinction, a place where the process of biological erasure is forced to stop. Saving the biosphere's most fragile species now requires moving them out of the natural environment and stepping completely outside the flow of time. In the center of this sterile neon blue laboratory sits a single ghost orchid. It is held in a state of molecular stasis, suspended inside a specialized glass chamber. In nature, decay is a byproduct of kinetic energy. Atoms constantly bump into each other, breaking chemical bonds and succumbing to the heat of the sun. Standard freezing cannot stop this. When a plant freezes, the water inside forms jagged ice crystals. These crystals expand and pierce through the delicate cell walls, the very blueprints of evolution. This creates a central engineering paradox. The only way to pause the plant's biological clock is to freeze it, but the formation of ice crystals physically shatters the cellular structures we are trying to save. Handling these specimens requires total environmental control. Scientists work in pressurized suits to maintain the precise conditions of the laboratory. The process begins with the extraction of microscopic pollen and tissue samples. Technicians use surgical microneedles to isolate these fragments without damaging the parent flower. The thermal stakes are high. Within this chamber, even a single degree of temperature fluctuation can trigger the chemical reactions that drive decay. Because a minor shift in heat could trigger 1 billion years of degradation in seconds, the extraction is a high-precision procedure where any error leads to specimen failure. To bypass the problem of deadly ice crystals, geneticists use a molecular loophole that alters the plant's cellular chemistry. Cellular water is drained out and replaced with a proprietary synthetic glass, occupying the cell without the chemical properties of water. As temperatures drop, this glass solidifies smoothly. Since no ice crystals form, delicate cell walls remain intact and protected. This shimmering, vitrified state is known as vitrification. The plant is no longer aging, yet its biological structure remains perfectly organized. By stabilizing the cell at the molecular level, scientists are effectively archiving the physical data of the species for the long term. Once the cells are vitrified, the specimen is submerged in liquid nitrogen vapor. This locks the orchid into its current crystalline state. The temperature inside this chamber is brought down to exactly -196 degrees Celsius. At this temperature, kinetic energy stops. Atoms no longer collide or break bonds. The chemical reactions that drive biological extinction simply cease to function. By halting molecular movement, the orchid is removed from the timeline of decay and archived in a genetic time capsule. The orchid's final destination is the Vault of Echoes. It is a silent forest, a massive archive of species held in glowing cylinders. Most of the plants stored here no longer exist in the wild. This room holds the remaining physical evidence of a lost biosphere. Dr. Eris Thorne describes the staff as librarians of the biosphere, curating a garden of species that are waiting for a future world that is ready for them again. The success of this project isn't measured in seasons. The digital readouts on these chambers count time in millennia. While the planet outside struggles to breathe, the heart of evolution continues to beat inside this lab, slowly, silently, and eternally. The biological clock has been paused.